We have got all the drug discovery functions that are needed to create, design, make, test, and develop drugs for HIV. It's a tough virus, it's a challenging virus, it's a sneaky virus because it knows how to integrate, mutate, and hide.
Will there be a cure for HIV?
And since the formation of ViiV Healthcare in 2009, we have been at the forefront of HIV research.
Thanks for joining us for this CBIA BizCast powered by Google. I'm Paul Pescatello, CBIA Senior Counsel and Executive Director of the Connecticut Bioscience Growth Council. In this episode, we're speaking with Dr. Umesh Hanumegowda. Umesh heads drug discovery at ViiV Healthcare in Branford. The ViiV Branford site is where the most significant research into HIV is being conducted, not just in the US, but really around the entire world. ViiV is a leader in HIV R&D, both for preventing HIV infection and for treating HIV. Umesh, thanks so much for this opportunity to learn more about ViiV and the cutting-edge research that's going on right now in the Branford labs. To start, could you give us a bit of history? How did ViiV get started? How did the company become a leader in HIV antiviral research?
Thanks, Paul and CBIA, for this opportunity to talk about ViiV Healthcare and all the great research that we do right here in Branford, Connecticut. ViiV's history, looking back at ViiV's history, it starts with the legacy of GSK's work in HIV. AZT was the first drug that was approved for treatment of HIV in mid-1980s, and that was developed by Burroughs Wellcome, which is a legacy GSK company. And soon after that, there were drugs like abacavir and 3TC that were also developed by GSK. GSK decided to create a company dedicated for HIV research and joined hands with Pfizer to start ViiV Healthcare in 2009. And soon after that, in a few years, in 2013, Shionogi joined ViiV Healthcare with their pipeline and became a shareholder in ViiV Healthcare. And in 2016, ViiV Healthcare brought Bristol Myers Squibb's HIV pipeline, and along with that, decided to start this state-of-the-art research facility right here in Branford, Connecticut, to do research in HIV. And since the formation of ViiV Healthcare in 2009, we have been at the forefront of HIV research. We have been the first one to introduce the second generation integrase inhibitor, which is the backbone of HIV treatment. We were the first ones to introduce the 2-drug regimen for HIV treatment. Remember, HIV had to be treated with multiple medications, and 3-drug regimen was the standard of care for a very long time. And with the 2-drug regimen, we brought down the drug burden and also the side effects associated with 3 drugs. And we were also the first one to introduce the attachment inhibitor for heavily treatment-experienced patients who have failed all other options. And we were also the first one to introduce the long-acting injectable regimen for treatment, for HIV treatment. And we were also lately the first one to introduce the long-acting injectable for prevention, HIV prevention. So I'm very proud to say that we have been the innovators and disruptors in HIV research and care.
You certainly have. I mean, so going back when the early days of HIV treatment, people talked about a drug cocktail, and patients would take upwards of scores of pills multiple times during the day.
Exactly.
And that's sort of what you brought to the table, is bringing that way down to.
Absolutely. Now it's 2 drugs and even 1 injection every other month.
This is amazing. Yeah. Does ViiV manufacture in Connecticut?
No, we do not manufacture ViiV's drugs in Connecticut, but we discover drugs of the future for HIV treatment, prevention, and potentially cure. So right here, We have got all the drug discovery functions that are needed to create, design, make, test, and develop drugs for HIV.
So, Umesh, what was your path to ViiV? I mean, you've been in the pharmaceutical industry for 20 years. What drove you here to ViiV and to focus so much on HIV?
So I started my pharmaceutical career in infectious diseases at Bristol Myers Squibb, right here in Connecticut. And my first program was to find an improved version of efavirenz. Efavirenz is an older HIV drug. And over time, I took on many other HIV programs, and gradually I developed a deep interest in the science of HIV. And in 2016, when ViiV Healthcare brought in the BMS's pipeline, I moved to ViiV and decided to stay focused in the area for a couple of reasons. One is we have the state-of-the-art research lab right here to do research in HIV. And HIV is challenging, and I like a tough challenge. And second, HIV disproportionately affects particular communities. There is a socio-economic aspect to the disease. There is deep stigma associated with HIV. So I feel that this is the population that I can help. And it makes perfect sense, right? Now we have this dedicated research facility here to do work on HIV. And with all the experience and expertise that I have gained over all these years, and the team that's here who have gained so much of experience over years in HIV, makes sense to put it in a place where we can succeed. So I want to be part of that success story of ending the HIV epidemic.
Well, that's a laudable goal. Just to back up a little bit, so is it safe to say that really the fount of HIV knowledge and the original insight into how HIV works was Yale, Bristol Myers Squibb. It was here in Connecticut.
It was. It all started here. There were quite a few research done here, but elsewhere across the world, other companies and NIH, of course, did a lot of work as well.
So, as you said, like HIV is a challenging virus. But just to back up a little bit, I think a lot of people are confused about the difference between a bacterial infection and a viral infection, and the two are very different. But if you could just give us a little bit of a primer on the difference.
Excellent question. And you are right. Both are infectious diseases, bacterial and viral. In the outset, they look similar, but knowing the cause of disease helps in picking the right type of treatment. So bacteria are single-celled organisms. They are living organisms. They are self-contained, and they have all the cellular machinery needed for surviving and to multiply. And they can survive outside the host. If the environment is conducive and they have got enough nutrients there, they can survive, they can multiply. And once they get inside the host, they can survive, again, they can multiply, and they can cause diseases like strep throat, food poisoning by salmonella, Lyme's disease. All these can be treated with antibiotics, and there are several different types of antibiotics. Virus, on the other hand, is tiny. It's tinier than bacteria. So tiny that it can even infect bacteria, and they are called bacteriophage. And unlike bacteria, virus...
Is it something like 1/10,000 smaller than a bacteria?
Not that smaller, but small enough that they can infect the bacteria. Unlike bacteria, virus is not a living organism. It does not have life on its own. It is only a package of genetic information and some proteins. But an intelligent package that once it gets inside the host, it can come to life. And it uses the host's cellular machinery to survive, to multiply, and they can make copies of itself, and they can cause diseases like common cold, COVID, hepatitis, HIV, and many more. And all these can be treated.
And all the different viruses you mentioned, I mean, they're all very different. HIV is particularly challenging. Why is that so?
Absolutely. So to answer your question, why HIV is challenging, we need to understand a bit more about the virus, what the virus does at a molecular level. HIV, once it gets inside the body, it specifically targets a type of blood cells called lymphocytes. And once it gets inside the lymphocytes, it can insert its genetic information into the host's genetic information. And once it does that, it can very efficiently make many, many copies of itself. It can just make copies of itself to an extent that the host cell dies, and the newly formed copies can then infect other healthy lymphocytes. And this cycle goes on. Unfortunately, the type of cells, the lymphocytes that the HIV infects, is integral to mount an immune response for any infection. So it's like targeting the soldiers specifically. So now the host cannot fight infections like a normal, healthy individual would do. And additionally, the virus can mutate. What that means is it can change. And these changes are very subtle, but significant enough that the virus can become resistant to the antiviral medication. And lastly, the virus can hide. It can stay latent in the infected cell and escape immune surveillance, but it can resurface if the treatment is stopped. So it's a tough virus, it's a challenging virus, it's a sneaky virus because it knows how to integrate, mutate, and hide.
Yeah, quite challenging. So how do your drugs combat that?
So we have got many options for treatment right now. You know, as I said, we were the first ones to introduce the 2-drug regimen, and we have got medications like this long-acting injectable, again, very effective. Drugs like the attachment inhibitors that can be given to heavily treatment-experienced patients who have failed other treatment options. And for those who are at risk of getting infected, there are these pre-exposure prophylactic medicines.
Yeah, if you could explain that a little bit. So there are several ways to combat HIV, both pre-exposure and post-exposure. If you could just explain for us the difference between the two strategies.
All antiviral agents can work for both, but there is one main difference between pre-exposure and the medicines used for treatment. For pre-exposure, these can be single agents. These are single class of drugs, but they should be strong enough that they should have the broad spectrum of activity to provide the protection. While for treatment, it needs to be more than one drug. So a minimum of 2 drugs, which we have demonstrated, is quite effective, and it could be taken as an oral medication or an injectable medication.
So how have HIV medications evolved since the 1980s? Again, back to the question of were there multiple medications? Because you were just saying that there are 2 medications.
We have come a long way. To your point, yes, there were days where people had to take multiple medications, handful of medications, multiple times a day, every day. And from there, we are here where we can combat with just 2 medications and infrequently. The HIV virus can mutate. And once they mutate, they can become resistant to the HIV, a class of HIV drugs. So there is a lot of research that has gone into finding newer classes of drugs that can affect different stages of the life cycle of the virus. Like, for example, we have got attachment inhibitors. fusion inhibitors. We have got broadly neutralizing antibodies, reverse transcriptase inhibitors, integrase inhibitors, protease inhibitors, assembly and maturation inhibitors, to name a few.
So it's a moving target. You're essentially always adjusting.
Exactly. We need to be one step ahead of the virus.
Absolutely. So, some HIV patients are, the drugs work so well, their viral load is described as non-detectable. So tell us about that. And is that essentially a cure?
Interesting question. You're absolutely right. We have got great medicines now, and especially when we consider the long-acting injectable, where one don't have to worry about missing their daily oral meds, it almost feels like you are somewhat, free of that burden, right? So somewhat it could feel that you are getting close towards the cure, right? And there are daily oral meds which are perfectly effective, but irrespective of whether it is a daily oral med or injectable, once a person adheres to their medications, their viral load in their blood becomes undetectable. And once it's undetectable, they cannot transmit the virus to others. And that's called U equals U, which means undetectable equals untransmissible. And that's the key message we strive to deliver, because I believe that will significantly reduce the stigma around HIV.
So is imaging a big part of what goes on in your lab? So, often I hear scientists talk about that laymen don't understand that just understanding the three-dimensional surface of a virus is so critical to understanding how it works and finding treatments and cures. Does that jive with?
Yeah, absolutely. That's any kind of three-dimensional imaging all the way, because viruses, they need to be, you cannot visualize by regular microscopy. You have to go into a scanning electron microscopy, so tiny. So yes, imaging is absolutely important, not just from a virus perspective, even from, for example, a reservoir, what we call where the HIV can hide in the body. And these could be like tiny tissues in the body where we can label, the targets with the appropriate dyes, and we can do this imaging. All these are very helpful.
Just back to the antiviral research for HIV and how it's informed other antiviral work. So has it affected COVID, certainly in the hepatitis space? If you could just speak a little bit about that.
Again, a great question. So yes, there are many areas where the knowledge that we have gained from HIV research has helped develop medicine. And as you said, the greatest example is our response to the COVID pandemic. The infrastructure that was built during the last 4 decades of doing the HIV research played a tremendous role in our response to COVID. By infrastructure, I mean the research infrastructure, the clinical trial network, the network of institutions and agencies that provided access to the HIV meds. They all played a critical role in our response to COVID. The diagnostics, the monoclonal antibodies, and ultimately the vaccines that were made for COVID all came from a good amount of learning from HIV research. The clinical trial network facilitated the rapid conduct of the clinical trials, and the global network of agencies helped getting those vaccines to the people, and thus helped end the COVID pandemic.
So will there be a cure for HIV at some point? And what form will it take? Will it be a vaccine? Will it be a post-exposure drug?
That's our goal, to find a cure. And from a vaccine point of view, we are looking for a therapeutic vaccine which could be given to people living with HIV to achieve long-term remission or long-term drug-free remission, and also reduce their reservoir size and over time cure them of HIV. When we talk about curing HIV, there are two aspects to it. One is what we call as a functional cure, and the other one is what we call as a sterilizing cure. Functional cure means where we can achieve long-term drug-free remission. Essentially, one feels like they are functionally cured without the drug. The other one is the sterilizing cure. Now, what that means is removing every viral particle from every infected cell in the body, which is going to be challenging. But I am optimistic that we will have a functional cure. Now, regarding the question on what form that could be, it's not going to be as simple as a single tablet or a single injection that can cure. It has to be a combination of medicines and strategies. And these could come from multiple companies, big or small, biotechs, and academia.
So there's an initiative to end HIV by 2030. So several governors and federal administrations talking about signing on to it. And there's a business coalition to end HIV by 2030. How do we end HIV by 2030? Is it a cure, or is it a combination of new drugs and public health measures for testing, things like that?
Again, a great question. So yes, there is an initiative to end the HIV epidemic in the US by 2030. And that's by the US Department of Health and Human Services. And the goal there is to reduce infections by 90% by 2030 using 4 different strategies. One is diagnose as soon as possible all the people living with HIV, and then treat them as early as possible with the HIV medication. And third is to prevent new infections by using the pre-exposure prophylactic medicines. And lastly, to respond to any potential HIV outbreak areas with proper medications, either for treatment or for PrEP. UNAIDS also has, or it's coordinating efforts to end AIDS by 2030. And that's based on a few countries which have achieved the 95, 95, 95 targets. What that means is 95% of the people who are living with HIV are aware of their status, and 95% of those are getting their treatment, and 95% of those getting the treatment are virally suppressed. Now, globally, that's an ambitious goal, but we are getting closer.
It's a laudable goal. So hopefully we can sign on to it here in Connecticut. So what are the challenges in terms of, part of it is identifying persons at risk for HIV, ensuring that they're treated and that they stay on their medication. So how does that work?
Testing. Testing is a critical piece of ending the epidemic. Only when we expand the testing will we know who are living with HIV, and only then we can start them on treatment. And with the long-acting injectables, which improve the adherence, I think the treatment and adherence part of it will be addressed very well. So one is testing, treating, then retaining them on medications, especially on ones that has got better adherence, like this long-acting injectables. And in parallel, provide the PrEP options for those who are at risk of getting infected. I think that's the only way that we will be able to end the epidemic. Test, treat, retain, and provide PrEP.
Is it easy to get tested? Are there ethical issues about testing?
More than ethical issues, I believe it's the policies of getting this HIV testing done as a routine screening. As you go to the doctor's visit, get the testing done, and that should kind of become a standard practice. I think so.
So, in all this work you've done on HIV, you've learned a lot about viruses generally, and so how has that affected ViiV's future? Do you think that you'll segue into other viral research and medicines directed at there are a host of viruses out there?
We have a lot to do. We have to reach those 40 million people living with HIV, provide treatment to them, get new infections under control, provide the PrEP options for those who need them, who are at risk. And finally, we have to find a cure. So we have a lot to do. So at ViiV, we are going to stay focused in the area because we strongly believe that staying focused, and we have demonstrated that staying focused will fetch us the right results. So we are in it till HIV and AIDS aren't.
Excellent. So ViiV is founding member of the U.S. Business Action Coalition to End HIV. Why is it important to ViiV to bring additional corporate partners into the coalition? I should say that CBIA is signed on to be part of that coalition.
As we talked, and probably you alluded to in one of your questions previously, HIV epidemic, or any epidemic for that matter, cannot be ended by one company or one agency. It has to be a collective effort. It's a collective action of the researchers, the pharmaceutical companies, the government agencies, and most importantly, the community. So I think that is where bringing in additional corporate partners will be very helpful in supporting this collective effort to end the HIV epidemic. And ViiV has been started this coalition 3 years ago. And at that time, we had 15 partners sign up. And now over 3 years, we have grown into 60. And that speaks volumes. So that means the corporate leaders in the state, they do recognize that HIV is an issue, and they have voiced their support, and they have signed on to help achieve our mission of ending the HIV epidemic. And the ask is simple. There are multiple ways that they can contribute. It could be as simple as the leaders of the business publicly show their commitment and show their commitment across their platforms. And I'm hoping that they will reach out to the association for more information. So I'm hoping that many partners will join so that we can end the epidemic starting right here in Connecticut. If anybody is interested, they can go to the website healthaction.org for more information.
It's such a valuable and effective coalition, just in terms of education to employees and things like making testing more readily available and increasing people's knowledge about the availability of testing. So, yeah. Well, Umesh, thank you so much for letting us know a little bit more about ViiV and all the work you're doing on HIV. It's an incredible Connecticut story about all that's going on here in your lab. So thank you so much.
Thank you, Paul.
And thank you for listening to this week's BizCast. You can listen, like, and subscribe to our podcast wherever you get your podcasts. And please leave us a review and let us know if you have ideas for future podcasts. And for more episodes, please head over to CBIA.com.